I named this presentation Enigma of the interface, because there is really something we do not understand, or we try to avoid to understand. Now, in this figure, you may see some slide of the land in the neighborhood of a wood. And here you have some very interesting elements, namely some trees with their branches, or some plates of chalk, and some earth, and some other plants in different areas. So there is an interface with the air, an interface of these elements, the trees, with the air, and the chalk, and some other things we do not really know exactly, and some plants. Now the interface is defined as a sharp contact boundary between two substances, materials or phases, either or both of which may be solid, liquid, and gases. So this is what David Williams said for this definition. Here you see some gentlemen representing the Council of the European Society for Biomaterials from some countries in Europe. And for example, this is Sandra Dorns, and Mario Barbosa, and Jan Feyen, and Uwe Soltes, and Paolo Giusti, and the other one is Marcel Josefovich. I show this picture because it means that there must be collaboration to solve the problems of the interface and the behavior of biomaterials in general. And these persons represent different fields in material science and in medicine as well. Now, this Marcel Josefovich is a chemist, and he wrote a review about the situation for the biospecific molecular recognition in living systems is known to be based on the lock and key principle as proposed by Emil Fischer. Based on this concept, biospecific polymers have been produced synthetically by attaching biospecific keys to the polymer chain. Now, this is very important because this Emil Fischer in 1894 came to this lock and key principle, and he did it on the stereochemistry of sugars, and showed that only if the structure is compatible with the lock, they can be transformed in plant or animal bodies. He is one of the Nobel Prize winners, 1905. To put an implant in a body, a human body, living human body, is necessary to make a hole, at least. And therefore, it's necessary to know wound healing. All these processes that are connected to wound healing. And since my time is limited here, I decided to give you information for wound healing by a compilation of text and figures by some co-workers of Dr. Reichert of Duke University, and you see this article in the internet, and for your learning purpose, I have copied this information, which is freely available, and you can have this information on your system, your university system. Now, going back, this is, for example, a wound. A wound 4 weeks after colostoma was made, and here you see the extracellular space with the fibers and some fibrin and epithelia that are growing to cover this area. And there are many cells and some cells with different functions, and in fact we are not able to really understand how, for example, the basal membrane is made, and this is very important for the epithelia that they are growing, and they need some guidance, and so this is, for example, one thing that we need to understand if we like to insert implants that are perforating the surface of the skin, and there is a similar situation for perforation in the other organs covered maybe by epithelia in the stomach or somewhere else. Now, implants. I compiled some morphological data on my first 40 postmortems, and this was published in '68. Here you see a lead with silicone covered, and here there is the platinum covered electrode. And the question at that time was, what does happen to the lead and to the battery? And here you see there is new tissue around the electrode, and in the neighborhood you see some type of the myocytes of the heart. Now, at that time, it was not really clear why the heart cells, the myocytes, are in some way tolerating this permanent electric stimulus. And it was demonstrated and is nowadays regular processes for patients who are attained by pacing problems. Now, to take home, information is put here, and there is cell and tissue response and material response at the interface. And it is necessary to understand mechanisms of wound healing of cells and extracellular matrix, and the whole thing is only understood if there is the key and lock principles followed. This is essential for the attachment of chemical species. So here on the left side you see the disaster of an implant put into the femur of a patient this was collected in 81. And here you see a femur with a broken head, and there was some nail put in the head, and the head is partially necrotic. Here you see a nail going down through the interior of the femur. One plate, screws, another plate, cerclage. And of course, this is not tolerated by the bone. And you can clearly see in this macerated specimen that the bone is very pale in relation to the living areas here and on the proximal area. So this is a treatment or over-treatment and some attempt to save the limb and to get the patient to walk again. Nevertheless, some principles have not been observed. Of course, to put all this material to the surface means that the vasculature is ruined, more or less, and the outcome is really a disaster. On the other side, you see an implant, a more modern type of implant with a stem and some collar and metal head. And the whole thing is put in the femur, and you see also the technique to fix the stem by cement. Cement is, in this case, polymethyl methacrylate, so-called bone cement. And this cement is introduced in between the trabecular bone. And of course, we face the problems there, maybe not in the early stage after the implantation, but later on. And this is documented in these two figures, where you see the cemented area here in higher magnification, and the bone, trabeculae, and this is bone marrow, and here you see a discoloration of the bone. This is a technique where we make sawn sections, and the stain is from Cosa, stains the bone, mineralized area of the bone, and fuchsin, this is soft tissue in the neighborhood. And here you see the effect of this cement. The cement, this methyl methacrylate, is an acid substance, and this gives you here the explanation for this change in color and in mineral. Now this is a more advanced stage of such type of cemented stems and the interface between the cement, which is here, with the zirconium particles introduced to make the cement radio opaque. And here you see some rest of the cortical part of the femur, and in between soft tissue. Now this soft tissue is generated partially as a reaction to the area of the demineralized bone. Here you have these remains of the trabeculae, and the fact that here is something new, and the material we see here is some debris from the cement on one side, and especially the components of these zirconium particles. Now, there are also other possibilities to fix a stem without the usage of the cement. And here is an example of an uncemented implant of a femur, and here you see a cross-section of this implant. This implant is covered by 1 micrometer thick area of very small particles of titanium, commercially pure titanium, and it was in situ in this patient for less than 1 year. And here you see the cross-section of the cortical area, and a very important reduction in bone due to the unuse of the bone. So this bone is not loaded and unloaded as usual, because the load is transferred by this new developed trabeculae to the stem, and this is on the lateral side, on the area anterior and posterior, and the medial side. So in this area, the trabeculae clearly are able to sit here and to have such a picture of the three-dimensional structure of this trabeculae sitting here in the voids of the surface. Now this is more or less successful with a higher magnification of the whole thing, and important is to acknowledge that there is no trabecular attachment here in the lateral side, but only where you have mechanical pressure or decompression. And when we go to a higher magnification, with this von Cossa and Paragon stain, you see that in fact the newly established bone is mineralized in a regular way, and sitting here in contact to the surface of the titanium. On the other side, you see some of multinuclear cells, macrophages, some fibroblasts, and these macrophages contain some particles, small particles of the surface. So apparently the tissue, the bone tissue, is able to tolerate a number of particles, but not too many. We do not really know the threshold for this phenomenon. This is another case where we have an implant in the left humeroacromial joint. The replacement was 2 years ago, and the change of the prosthesis 3 months ago in an old person, and the picture was taken with polarized Nichols prisms. That means you have a birefringence of the particles, and you see this enormous amount of particles. Of course, this is a really big problem for tissue to function in a normal way. This is not possible. The macrophages are predominant in this situation, and they contain the particles. This is a immunohistochemical stain for macrophages with PGN1. Aberration plays an important role, for example, for these morsel caps, where you see the scratches here, and the generation of particles is unavoidable in this phase. And here you see, of course, these particles in the neighborhood of this implant. On the other side, if there are no particles, there may be bone in contact to this titanium surface. Now, we dealt some years ago with the problem of load of tissue by these different chemical species, iron, chromium, nickel, cobalt, in the neighborhood of implants. And here you see a compilation of our investigation with the number of cases, the maximal load, the minimal load, and the mean. And if you think that this can be tolerated, you are really on the wrong side. So particles influence cells and tissues, of course. Mechanical mismatch generate particles. The implanted stem is much less resilient than the bone, and this mismatch makes the interface in such a way that there are scratches, and the release of particles is not avoidable. There is, of course, also important the size of the particles, whether millimeter, micrometer, or nanometer, and the number of the particles. Some particles may be tolerated if a limit is not recognized. There is inflammation, particle-induced inflammation. And these particles can be transported to the regional lymph nodes, remote lymph nodes, spleen, bone, and even brain. So this is really relevant. And nanometer particles usually are not observed in the light microscope, but influence activity of inflammation in a not yet very well recognized matter. We dealt with the question of nanoparticles versus microparticles. And we did this in collaboration with one institute for testing of material in Berlin. And in comparison, where corundum with micrometer or nanometer size, chromium, nanometer, graphite, micrometer, and nano, meter size. And what I like to show is that in our cell culture experiment with macrophages of a derived type, we see in different abilities of the particles. So, for example, in distilled water, you have an important difference in the setup potential. This means the setup potential is the electrical possibility of the particle surface between minus 14 and plus 7. And if you look into the relative values for the graphite micrometer versus nanometer, there is a difference. And we use also the measurements of the setup potential when we introduced the particles into the cell culture medium, and there you have also important differences. That means that the particles have different possibilities to attract biological molecules and to change the molecules, maybe the conformation of proteins, and this makes an important point because these surfaces, these particles change all the time, and we did some also recent investigations and took some genes that are expressed by these macrophages. You know that macrophages usually are able to deal with microbes, and there are on the surface of these microbes some features that are FC receptors or complement receptors or scavenger receptors, other integrins, lectins, toll-like receptors, and so forth. And they have influence on as activating signals or inhibitory signals to the macrophages. And there is a list of receptors we also used for this gene profiling, and the result is very interesting because we saw, for example, for TNF alpha that this is a negative control, positive control, and this micrometer and nanometer corundum, aluminium oxide. And you see that the micrometer sized corundum particles have a decline in the reaction, in the production of TNF alpha, comparable to the negative controls, and for the different time 4, 24, 48, 72, and 96 hours, whereas there is an increase, a dramatic increase in the production of TNF alpha for the nanometer sized particles and the normal range, which is given here, is even after 96 hours increased up to 58-fold for mRNA and 10-fold for the protein. And there's a similar thing for the colony stimulating factor in this experiment. So in practice, we have to be very cautious with nanometer particles in the body. Now, I would like to come to another point, and this is bioactive materials. Bioactive materials were invented or found by Larry Hench. And here you see a picture of Larry Hench. He died unfortunately in December. This is Larry Hench. And Michael Sefton and June Wilson and Janas and Soltes. And this was at the occasion of the Third World Biomaterials Congress in Kyoto in '88. Now, this gentleman, Larry Hench, put the principle of bioactive materials in some books, and one of these books is recently published, and he divided the classes of bioactivity to osteoconductive or osteostimulative biomaterials and soluble cells and phosphorus species, calcium and sodium ions, influence transcription vectors and gene activation. And there are others that are connected to osteoconduction. Now, Wiedelt in the 70s with similar materials as the University of Florida, the Enigma material, UF45S5, with these materials, and I must say that I destroyed the application of these materials to surfaces of implants in Germany, and if you like, also in Europe. So implant stimulated interfaces were one of our meetings in Berlin in 1983, and here you see some of the people dealing with these materials, namely Thomas Albrechtsen from Göteborg in Sweden, Hans Planck from Vienna, and Volker Schulz from Berlin. The surfaces of these materials change. If you put them, for example, into some solution with pH 7, the ceramic part, the mineralized part, is dissolved earlier than the crystals. The crystalline part, as you see here, and this is more dramatically if you lower the implant solution, and here you see, for example, with a pH of 4, you change the surface importantly. So you find all the time different surface structure at implantation and after implantation, and the modification is, for example, by change of pH and fluid turnover, and this influences cellular activity. These classes of bioactive materials have the ability to cover the surface of implants very early, and after 60 days, for example, you see that up to 90%, 95% of the surface is mineralized bone. And of course, this was very interesting. However, if you look to the surfaces and to the surface, for example, of this bone bonding material, cervital, after 3 days you find macrophages. They change the surface. The macrophages display acid phosphatase, as you see here, and after 7 days there is already bone at the surface, so all these macrophages disappeared. Why they disappear and what they released in the time and at the surface is not yet clear. However, we know that the bone at the surface of materials with this other composition may be inhibited in mineralization by, and also in differentiation of cells. Here you see, for example, some cells that are not bone-like but more some other cartilage cells. And this is also true for the surface of calcium zirconium phosphate, where you observe that this area of the bone is not yet in regular way mineralized. So the mineralization processes play an important role. For example, you see here the mineralization of primary bone with matrix vesicles, and these matrix vesicles have special way to develop, and they can be seen in the young primary bone, and there is also another possibility, namely that the mineral of the implant, this is an organoapple by Sam Stubb, is in contact with the mineral of the collagen and the bone in the neighborhood of this material. So the implant interface changes by time in structure and chemistry. The cells and tissues that are not in use are absorbed and disappear. This is an important point. And the smallest particle are the most dangerous. So this would be something for a discussion. Now there is another, and this is my third part of the lecture. Inflammation. Here you see, for example, the opened left ventricle after implantation of aortic valve implant, and you see that the surface of the aortic valve is more or less destroyed by bacterial infection. And this is a really big problem in the clinical usage of materials, different materials. This is another example of an aortic replacement with a collagen material, and here again you see the bacteria and the patient died. Another implant-related problem here, the column, the vertebral column was broken, and there was the implantation of this titanium mesh and some fixation, and the whole thing was infected, and the patient died. So this is of course not the regular way. However, it must be recognized that we are dealing with multimorbid patients. They have cardiovascular diseases, for example, arteriosclerosis, stenosis, aneurysms, metabolic diseases, diabetes mellitus, gout, osteoporosis, osteoarthritis, spondyloarthropathy, chronic inflammation with chronic polyarthritis, pyelonephritis, glomerulonephritis, and so forth, trauma, bone fractures, tumors. So we are not dealing with simple systems. Of course, there are other applications for implants, for example, to replace parts of the aorta with dacron, and on the other side to implant stents. Stent, for example, for the cholecus, and this is only a very short-living possibility to release the patient from jaundice. And for example, here you have also additional damage of the tissue, in this case of the tumor, after implantation of the stent in the cholecus. Here you see the carcinoma that expanded from the pancreas. The problem is, especially for young people, the rupture of the cruciate ligament. These are mostly people doing sports very roughly. And for example, here you see the reaction around the carbon fiber replacement of the anterior cruciate ligament. Or this is another example for the PTFE, polytetrafluoroethylene, replacement of anterior cruciate ligament, where you see important inflammation and reaction in the neighborhood of this material, which is in some areas broken. Now, this is a higher magnification around one of these cruciate ligament replacements, where you see that there are collagen fibers in the surface or near the surface. And I always ask people to try to find ways to make a lateral attachment of artificial material with collagen fibers to simulate the attachment that is realized in ligaments. This is especially Sam Stubb from Northwestern University, who dealt with self-assembly, and this principle of self-assembly should be made for collagen fibers, membranes, fasciae, ligaments, and there are other candidates for this. I come to the end. Anthony Christina said years ago, there is a race of microorganisms to the surface, and this means primary infection and also in secondary infection. And of course, even if you have implanted material without contaminating the implantation bed, there can be intercurrent bacteria, and so loss of the implant. And the question is also, what is success or failure? Do we need animal experiments, or do we treat human patients without sufficient testing to avoid animal experiments? This is an important point for further developments. And I am of the opinion that we urgently need animal experiments. And the reason for this is that in vitro cell testing does not predict tissue behavior. We have unsolved problems. For example, collagen attachment, self-assembly of biomaterials in the extracellular matrix, attachment of epithelia for skin-penetrating implants. What does happen to nanoparticles and to people with nanoparticles in a living environment? We need some solution for nerve guide and nerve healing, and bacterial, viral, and fungal contamination. That means biofilm development should be in some way treated or avoided, and one problem is also drug resistance. So after my presentation, maybe there is a little bit more color in the whole field for you, and I come back. You can use this material for information and learning as you want, if it's loaded in your learning program. Thank you very much.